Basically, you have the form of life, and it evolves into two separate forms. Okay? How can you determine how long it took, number one, and which forms evolved from which forms? Well, you can't analyze the genome that they evolved from, because, of course, that's gone, unless we can do more work on ancient DNA. Otherwise, we really can't analyze, of course, the DNA of the original form. So you analyze the offspring, the two that came from the original form. You analyze the DNA of this offspring, and you compare the two. And the more differences there is between the two offspring, therefore, the longer it was, they evolved. Now, you probably see problems with this already. The longer time ago it evolved from the common form, okay, and the farther they are apart, okay, the genetic comparisons, not only the longer time span it evolved from the common form, but also the closer they are, okay, given how close the two DNA comparisons are. So if the two DNA comparisons are enormously different, then it evolved a longer time, okay, and possibly it evolved from some other form, some other common ancestor, okay? And the problems were recognized right away when this was proposed. And the goal is to develop the tree, the taxonomic tree, number one, and number two, how long ago the divergence occurred. So that's the two things they're basically looking at. And it assumes the taxonomic tree, something like this. This is an older picture, but it assumes this is true. So we're beginning, in fact, there's a lot of assumptions, which of course I couldn't buy at all. But nonetheless, you have to understand where they're coming from, how they get this philosophy. Okay, the second problem, well, let me just mention, it clearly has had a major influence in all biological disciplines. And I give the reference here. I have about 6 pages of references, so I did a lot of research. And I had to try to get this down to 50 minutes or so. And the basic assumption is DNA evolves at rates that are constant through time and for different lineages. Well, right there, I thought that's a problem, and others did too, which we'll talk about. And they use genetic comparisons to estimate the number of mutational differences. And of course, they assume, and this is one big problem, they assume that all the differences in the DNA are due to mutations, or to genetic drift, or other natural factors. So they assume that the original life form had a set of genes, somewhere way back a long time ago, 3.5 billion years ago, and then all the genetic differences we see today is a result of the accumulation of mutations, genetic drift, and other factors. Primarily mutations and genetic drift. So that's the assumption. Okay? You can think about that. Try to get your mind around that. That all the genetic differences we see today, every one of them, are a result of mutations. And the basic problem I had with that is, well, if you read John Sanford's work, the basic problem I had with that, and I teach this stuff, my medical biochemistry textbook that we use makes it very clear that each generation, there's 100 to 150, and these are estimates, of course, 100 to 150 new mutations in the gametes. So you have between 100 and 150, on the average, more mutations than your parents. Your children have between 100 and 150 more mutations than you do. Your grandkids have between 100 and 150 more mutations than your kids. Okay, and 99 point, and the numbers vary, but the most common number I come across is 99.99% of these are harmful or near neutral. Actually, most are near neutral. And near neutral means that there's no problem with 1 or 2, no problem with maybe 100, but they add up. Okay? It's like damage to your car. You have a problem with your transmission, doesn't work quite right, doesn't go into second, the engine knocks, the rings need replacing. Well, the problems build up, and each problem is not lethal to the car. But the problems build up, and then what happens? The car eventually doesn't run. In fact, the best example of that is aging. What is aging? Aging is nothing more than the accumulation of mutations. Well, the same thing happens in the genome. as each generation we accumulate more and more mutations, what's going to happen? And I know when I first started to present these ideas, it was not very well accepted. And then fortunately, John Sanford came along and did a book on this and did a presentation today in this room, yesterday, on this topic. And he showed now, giving clearly empirical data, the fact is the genome is accumulating near neutral mutations, and eventually what you have is mutational failure, or what they call mutational meltdown, or mutational catastrophe. And the species become extinct. So we're going, and I tell my students, they talk about evolution, I say, of course I believe in evolution. I'm an evolutionist. In fact, I agree with Darwin in his 1871 book, where he talked about the descent of man. That's true, I agree with him. Man is descending, right? What he meant was the ascension of man. It's interesting he didn't say that, or the ascent of man. You know, he could have used some other phrase, but he used the descent of man. And yeah, that's true. It's interesting that Darwin is prophetic, and he, well, he didn't, of course, use the same term that way that I am, but, and thinking of that, and I've done a lot of work on that, I'm doing more work on that, and clearly, and this is in the textbook, this is not controversial, we bring out and talk about this in class, in my biology class, in my biochemistry, I'll be teaching biochemistry next semester, and it's clearly, we're going downhill. And of course, most of my students are medical students, and so therefore, that's useful to talk about, because guess what? Medicine helps reduce that downfall. We can extend the age that people live because we understand now what many of these mutations do, and we can try to deal with it. A lot of mutations cause metabolic problems. So what we do is we use supplements, which replaces what the metabolic system is supposed to do. So we can stave off death many times for many years, but we have to recognize that, and of course students like to hear this is great job security for us. Okay, because there's always new diseases. There's, the last 10 years, there's what, 30 new diseases that came up, or at least they're new to medical science. They may have existed somewhere else, but we didn't really analyze them. A lot of people get sick and die, and we don't do examinations to find out why. In the Western world, of course, we're more prone to do that, but not always, because it's expensive. So think about that now. It takes my students a while to comprehend that the fact is we're going downhill. And a lot of the students that have kids say, yeah, it's true, I see that in my kids. They're clearly, they're going downhill. Okay, so this is what we're trying to do in this genetic clock. We're trying to compare differences and then estimate when they evolved from the common ancestor and how close they are. And the hope was that we finally have, and this is a quote, all these statements in here just about are quotes. We hope we have finally a scientific evolutionary tree of life. That was the goal. And a lot of people were very optimistic when this started. And the first problem with this has been presented earlier by Jeffrey Tompkins. They do all these estimates on gene conservation with humans, so they compare a mouse with humans and chicken with humans. And if you, hopefully you went to his talks and you're aware of all the problems with this. Okay? And right here is a big problem, 99.5% similarity between us and chimps, and of course the current number is what?
75.
Closer to 70, 75. Depends on the genes you look at. If you look at coding genes, exons, you get closer to 80, 89, 87. And if you look at other genes, other gene pairs, you get closer to 70%. So that's way off. And one thing I can mention here is it takes years for the new information. And this isn't just done by creationists, this is done by evolutionists as well. In fact, several of the papers we did, we just surveyed what evolutionists found. Okay? But it takes years for this stuff to filter down. And you still see this 99.5 or more often 98 point whatever. You still see that number all over the place. It still is commonly presented. And it's going to take years for that to dissipate. Probably the best example is when I teach anatomy, it brings out their no vestigial organs. The appendix is 5 different functions, and they list them. We talk about them. It's part of the course. You have to know what the organs do. Okay, then they go to the biology class, and the biology book says we got lots of vestigial organs, the tonsils, the appendix, the pineal gland, and a whole bunch of others. Okay? And they sit here and they say, now wait a minute, we just learned in biology that all these organs are vestigial. Now you're telling us they have a function. And I say, well, there's a good lesson from that, and that is that textbooks are not infallible. They make mistakes. And that's something that students, they want. They want their teachers to be infallible. They want to trust what we say. And they also want the textbooks, of course, to be infallible, or at least be able to trust it. So I can usually make that into a good learning experience. Okay, so the problem with genetic comparisons is that genomes we compare by and large today is from extinct life, from living life. That is the source of the comparisons. The only way to do this accurately is to make comparisons between life forms that have the same evolutionary age. So in other words, we have to go back 1 million years, evaluate the genome of some life form, and then look at their offspring, evaluate their genome, look at their offspring, and evaluate their genome. We need a line from 1 million years ago, at least 4, 5, 6, 7 generations, so we can look at the genomic changes that we see. And I'll show you a picture of that in a second. But basically, all we can do is evaluate the tips of the evolutionary tree. And that means that our first common ancestors, according to evolutionists, lived 3.5 billion years ago. Okay? Bacteria, E. coli, we use a lot of E. coli and Escherichia coli in the lab, and other bacteria when I teach microbiology. And how far back does their evolutionary tree go? 3.5 billion years. So all living creatures today, they go back to the base of the tree according to evolution, so they're all 3.5 billion years old. So we have 3.5 billion years evolution behind us, and E. coli has 3.5 billion years behind them. So all we can do is look at the tips of the tree. Gould stresses this a lot. He does a good job on a lot of his work. So when we examine the genome to determine the evolutionary clock, we must, to be accurate, we must utilize verified evolutionary ancestors, and we can't really achieve that. Okay, and so there's the beginning of the tree, and this is at the tips. They show this is distorted, but they're all the same age. They all stem back to this first single or several forms of life. And then from the comparisons of the genome, we get all these differences here. And I'll show you some problems. So when they first started doing this, actually they ended up with a pretty good way of comparing it. They used alpha and beta hemoglobin chains of different species. And they assumed one change equals 1,000 years. Now they could have assumed 3,000 or 10,000. These are somewhat arbitrary. These are original numbers. So, and I'll go through this. This is directly what how they reason, so you understand where they're coming from. Using the estimated time of divergence of horses and humans, 130 million years, and of course, how do they know that? Well, that's the estimated time. And the number of differences between the alpha change in the hemoglobin. They calculate the time about 14.5 million years for each amino acid change. And Tucker, Landi, and Pauling. Linus Pauling, by the way, was very involved in this. Kind of interesting how he got involved in this. He was involved in a lot of other things. He was a biochemist, got the Nobel Prize for his work in chemistry. And then his second wife was very concerned about atomic testing atomic weapons. And they realized that the testing the atomic weapons gave off huge amounts of radiation. They realized radiation causes mutations. So he basically started in this by analyzing genes to look at the mutational load, because he predicted the mutational load would increase because the increased amount of radioactive materials in the atmosphere as a result of atomic bomb testing. So he became very militant in this, very involved. He got a second Nobel Prize for Peace for doing this work. Well, in doing this work, he and his student, Zucker Kandi, I guess you pronounce his name, I think it's from Germany, but they used 14.5 million years as the standard rate of one change of each globin chain. The number of differences in the descendants' hemoglobin chain was used to estimate the last common ancestor of not only this, what he was looking at, but other animals. And a lot of the work was done with alpha and beta chains because they did a lot of work previously with that. So he used that because that was well known. So the amino acid analysis, and you can of course analyze the gene or amino acids, analysis of gorilla alpha and beta chains indicated the human and gorilla alpha chains differed only by 2. Later on they found this to be 1 out of 141 amino acids. Now, problem here is polymorphisms. If you analyze your gene and my gene, you're going to find differences, a number of differences. And how much? 10 minutes left. Oh my. So if you analyze your genes and my genes, you're going to find differences. You find lots of differences between all people. So you'll get a different molecular date if you analyze me compared to somebody else. So that's another problem. I'll go through this relatively quickly. And anyways, it's multiplying, dividing, and so on. And they got 11 million years difference, which close to the limit estimated by paleontologists. So they got a good start. But it's basically within a matter of, well, when they published this paper, the two prominent vocal skeptics were Ernst Mayr and George Gaylord Simpson. Probably the most leading evolutionists, well, at that time and still today. I've got probably 10 books by George Gaylord Simpson. In college, I had to read several of his books on evolution. So, but the assumption was, they assumed that molecules accumulate differences at a regular rate, and of course, it's purely a function of, well, time, and that turned out to be not true at all. And I'll show you some examples. The problem is, of course, you give different answers to questions about the time in a past events because you get different numbers, depends upon what you evaluate. To summarize what the theory is, one change per 1,000 years occurred, and that was decided the clock standard. So if you have 4 changes, 4 differences between the 2 comparisons you made, therefore you have 4,000 years separate them from the common ancestor. That was the theory. And it seemed to be successful at first, so there was a lot of molecular comparisons. In fact, many people still accept it. But the criticism is enormous. It's absolutely enormous. And I'll go through this. The goal was find the true evolutionary relationship of all living things. And this is done by Cornell, PhD. She did a study on this. And so everything was fair game. It didn't matter what you look like. It didn't matter. You just compared the genomic sequences, and that then was to produce the evolutionary tree. Okay? The problem is, though, we had a number, there's wide differences often existed between phylogenetic trees based on the fossil record. So you look at the fossil record and you get one evolutionary tree. You look at the genome and you get very different evolutionary trees. And that was, I'll skip all this. These are some of the problems with it. Basically, molecular studies generated widely divergent results. And one study, for example, they found a difference of around 400 million years for the same gene between the two. And looking at the phylogenetic analysis, there's no way there could be that much. And then you get some more interesting ones. One common ancestor of animals, this is Zun, I guess you pronounce that. One common ancestor of animals was anywhere between 100 million and 1.5 billion years before the Cambrian explosion using this technique. And they became more and more of these problems. Some molecular clock studies even place a common ancestor of the animals after the Cambrian explosion. Of course, that can't be true. And so, the one researcher, this I quoted him quite a bit. He was very critical, and I didn't want to load this up with too many quotes, but he said, these molecular clock estimates look deceptively precise, but given the many problems with this technique, the advice to readers is whenever you see a time estimate in the evolutionary literature, demand uncertainty. Okay? A little difference exists in the histone protein. This is because they're evolutionary conserved. So if you look at the histone protein, these are the proteins that the DNA is wrapped around. If you look at the histine proteins, you find that they're almost identical in all life forms that have chromosomes. Okay? So therefore, we don't use histines as part of the molecular clock because, well, you're always going to get a divergence of time because the histones are virtually identical in all life forms that utilize chromosomes. Another problem was we have ultra-conserved elements, and there's lots of them. They're 100 or 99 or close to that percent identical in a wide variety of life forms. So obviously you can't use this. So to some degree what we're doing is we're cherry picking. We're throwing out the ones that give us very, very old dates, going back 1.5 billion years, and we're throwing out the ones that give us very new dates. 5 minutes, okay. See if I can get some summary. And there are lots of highly conserved genes, monosaccharide sequence that are by glycosamine, heparin, and so on, and especially enzymes. They seem to be conserved. And so scientists therefore have to cherry pick. And whenever you select your data, you got a problem. You have a problem. And we know the changes vary enormously. Because some are highly conserved, some are less conserved, some are not conserved at all. So when you cherry pick your data, it becomes problematic. And you can see a good example. Depends on what gene we use, we get these different parts of the evolutionary tree. They can be arranged all over the place. And inaccurate and inexact calibration standard, and so therefore to produce an exhaustive evolutionary timeline that is enticing but totally imaginary, and they concluded that this is the molecular equivalent of the Usher creation of the Earth on October 23rd, 404 BCD. So if you want to put somebody down, just quote the Bible and that really will put them down. And these dates, these authors note, have been mass-produced in the most prestigious biological journals. And this is the problem that you have with so much of this stuff. Once something gets out there and spread, people accept it as true because we hear it so many times. I read about a book a week on the average, and I watch a lot of films, science films. I've seen all of the Neil deGrasse Tyson films, and you see this stuff repeated over and over and over again, and you accept it as true because, well, everybody, quote, knows it's true. And to some degree, that's what has happened here. They're repeating all kinds of dates in major biological journals, and therefore it becomes accepted, not because there's evidence, but simply because one hears it over and over again. Cytochrome C, probably the most famous example of that. Gish, in a lot of his debates, had to do with cytochrome C. Cytochrome C probably is one of the best ways of determining these molecular clocks, but again, we're cherry picking. Hotspots, a big problem. You can see that the mutations is not equal across the genome. This is E. coli. And here you can see this is 1, 1, 0. This is analysis of a large number of genes of E. coli. Here we got 2, here we got 3, and here this is a hotspot. And hotspots are interesting because often we get back mutations there. It mutates back to the original. It goes back and forth. So it's called back mutations. And then they looked at a set of salamanders. They looked identical to one another. As far as they could tell, morphologically they were, but they were obviously, from looking at their physical structures, members of a single species, but yet when they did DNA analysis, they found that two salamanders were entirely distinct, even though the two types were separated by only a valley. So DNA analysis says they're very, very different. Best example of this, and skip on. Well, this is important. We used to think that a gene started here and ended here and all done. It's got the AUG, start codon. It's got the stop codons, UAA, UGA, and UAG. And that's it. Now we know they're genes within a gene, they're genes that overlap, now we know it's a mess. And we also know that 94% of human genes generate, according to one study, more than one product. And a good example of this cam, okay, there are 95 alternative spliced exons, so you get 38,000 possible isoforms, 38,000 different proteins from this one gene. So you may have two genes which are identical, and your molecular clock would say they evolved very recently from the common ancestor. But yet, in fact, the two genes, even though they're identical, they may end up producing very, very different proteins. So if you compare proteins, you get one date. If you compare genes, you get another date. And a few more examples. I want to leave. An orphan gene, that's a big problem. Orphan genes are found in one organism and no other known organism. Now, as we sequence more and more life forms, we may find similar genes to the orphan genes. But as far as we know, orphan genes are found just in one organism, and that's it. And they estimate 10 to 20% of all life forms, of all the genes in each life form, 10 to 20% are orphan genes. Humans, they estimate from 10 to 30% of all proteins are produced by orphan genes in humans, at least according to the data we have now. So these genes have no evolutionary history. And they explain them by massive mutations. Okay? And we talked earlier, the other speakers, incomplete lineage sorting. This is a big, big problem with this theory. And the biggest problem, well, we got contamination. That's a problem which the sequencing should be done in clean labs. But most of it is not done in clean labs. And we have interesting differences. A crocodile alpha hemoglobin shares only 5.6 similarity with vipers, which are also reptiles, but 17.5% similarity with chickens. So crocodiles, according to this, are evolutionary closer to chickens than they are to vipers. And I'll give you one last example. This is archaebacteria and eubacteria, or true bacteria. These right here, morphologically, at least externally, they look similar. The coloration, of course, is artificial. But under a microscope, we don't have any archaebacteria in my lab, so we've got lots of eubacteria. But if we did, we would see they're almost identical, morphologically, at least externally. But yet they're so different that now we have three domains of life. Bacteria is one domain, archaea is in another domain, and eukaryota, which includes plants and animals and reptiles and people and slime molds and yeast, all of this is in another domain. So this to me is a huge problem because these are all basically bacteria. These archaebacteria, though, have very unique traits. They can live in very high temperatures. Some are thermophilic, etc. So this is what I feel a huge problem with basing time as well as evolutionary trees on genetic analysis. And interestingly, there's only three types of archaebacteria: methogens, halophiles, and thermophiles, which is interesting. Okay, I'll get the last. In short, and I like his quote, empirical research shows molecular clock presents an illusion of precision that is not unlike reading the entrails of chickens. And many of the early doubts which we talked about still persist, and there are many others. In fact, the genetic talks that I went to here, as I listened to them, I thought of this paper, and I thought, here's another big problem, here's another big problem, here's another big problem. So my question is, there are so many problems, it's hard for me to understand why people give this any credibility. Now, maybe you can help me overcome some of my enormous doubts, but we'll see. So any negative comments, that's how I learn. When I send papers out for review, if they send it back and say, great paper, I loved it, I don't send my papers to them anymore. I don't want to hear that. The better job they do, the more they can find wrong. Because every paper I've written has something wrong. Right? And every paper you've written has something wrong, or something that could be improved. So if they find what's wrong, and then I can improve it, I'd rather have my friends find my mistakes or suggestions to improve than my enemies. So I find I only send my papers to people who really get into it, really dig into it, and say, oh, this isn't quite right, or you need to add something here. That's how I improve it. So, word to the wise. Thank you very much.